many of you think it's a 2n equals 14 cell? 2n equals 2. 2n equals 10. 2n equals 4. 2n equals 8. 2n equals 6. Okay, so what are the things you look for to try to figure out what the number is for this cell? Colors will throw you off, all right? Because it's a little bit of a different scheme than what we used. But what key things are being illustrated here and here? What are those? I hear lots of words that are related to it, but not the word that I'm looking for. We want a little bit more sophisticated language to that. That's good, all right? Whatever those chromosomes are called. Homologous chromosomes? Yeah. Okay. So are these homologous to each other? So is like the green one here homologous to the green one here?
No. Those are...
Good. They're not homologous, right? These are separate chromosomes, all right? So this is a homologous pair with sister chromatids. This is a homologous pair with sister chromatids. So it is a 2n equals 4 cell. And these, what do we call these? Starts with a T, E, T, tetrad, all right? So these are your tetrads. Okay, so that's one way when you look at a cell to figure out what's going on with the chromosomes. Is if there's a tetrad forming, that tells you something about the diploid number of the organism. Okay. So if we go to the very end of this, so if 2n equals 4, n equals 2, and so do your gametes, your haploid cells at the end of this, do they match your expectations for meiosis for a 2n equals 4 organism? Okay. They should, right? Each one has one big chromosome and one small chromosome. One big, one small, one big, one small, one big, one small. So they each have 2 different chromosomes. So if you were to fuse 2 cells with that, you'd end up with a diploid cell, or 2n equals 4. Okay. Did this confuse or clarify, or are you indifferent? All right, so we need some practice using a little bit of this terminology, and then also just thinking critically about what's happening to the information within the cell. Okay? We're not going to have you guys sketch complex. Just get through this. Okay. So one of the things that was a little bit dishonest about our approach of drawing meiosis was when you get to the end of meiosis for humans, for mammals, you don't actually produce in the female 4 eggs. You produce 1 egg and 3 nuclei that have genetic information in them but aren't actually involved in fertilization. So when you actually look at a human meiosis, like for a to produce an egg, you don't take it to the end and make 4 eggs. You make 1 egg and 3 little supplemental nuclei that basically are kind of shunted off to the side. And they're referred to as polar bodies. But I want you to notice that the products of meiosis are 4 haploid cells. It just happens that we put all of our eggs in one basket, so to speak. All right? We put all of the energy of the egg, of the original cell, into one cell, because that cell needs to divide a bunch of times before it actually implants and starts getting nutrients from the mother. And so you actually end up with 1 egg and then 3 haploid cells that aren't involved directly in fertilization. Yeah?
What do they, or getting there, what do they do? Like, what's their purpose?
They can, I'm not sure if the polar bodies in, there are some weird cases in other organisms where you get some of these supplemental nuclei fusing and making things, But in us, it's basically a way to do meiosis, as far as, unless you guys can correct me if this doesn't seem correct, but do meiosis to make a haploid cell, all right? But because we want to put all of our energy into one cell, so it has energy to divide enough times that it can actually become an embryo, all right? But the only way to get to a single cell that's haploid is by going through meiosis and making 4 cells. So we basically, their job is just to take the extra nuclei and leave all of the contents of the cell to one nucleus, if that makes sense.
Oh, it does. It's just how interesting.
It just seems weird.
Yeah, it's like,
So,
like an appendix. Maybe that's what it's called.
Yeah. Okay. So when you think about meiosis in humans for the sex chromosomes, you can, and you guys sketched this out, I think, for males on the lab that we finished up on Monday. If we go through male meiosis, and hopefully this looks somewhat like what you did, maybe with a fewer steps than what you guys actually sketched out. You have your diploid cell with an X and a Y. You make your sister chromatids, right? Then you line them up in a tetrad. But unlike other homologous pairs of chromosomes, the X and the Y don't recombine with each other. They line up as a tetrad, but they don't recombine. Then your pair of sister chromatids segregates off onto one side of the cell. Your other pair of sister chromatids segregates off. The cell divides, giving you 2 cells that have 2 X's or 2 Y's, who then enter into meiosis 2, line up in the center, and separate, giving you 4 cells that are X, X, Y, or Y. So hopefully what you got when you finished sketching out meiosis for the sex chromosomes were 4 cells where there's a 50% chance the cell has an X and a 50% chance the cell has a Y. And you contrast that with female meiosis, which you didn't sketch, which I've got drawn up here for you. And in female meiosis, you've got 2 X chromosomes. You make your sister chromatids for each. You line them up as a tetrad. And they can homologously recombine. The X chromosomes are able to. All right. Then you enter into metaphase and anaphase, and they separate. The cell divides, and they separate again, giving you 4 different cells that, regardless of which one actually becomes the egg, is going to carry an X chromosome. So the female always, barring some sort of error during meiosis, contributes an X. The male has a 50-50 shot at getting an X or a Y. And so if you randomly pull a sperm from a male and randomly pull an egg from a female, that gives you a 50-50 shot of having a boy or a girl. And that's why the sex ratio is so close to 50-50 for us. It's not exactly 50-50, but it's really, really close to 50-50.